Welcome to Synapsis. I'm Gökçe, a biomedical scientist specialized in neuroscience. And today I'm trying to take on a topic that's not the easiest to cover fairly, and that's grief. Whether for family members, friends, lovers, or animals, perhaps for some even nature's other elements. The only condition for grief is not just the sense of ownership, but really it's love. If we love, we grieve. It's a pact. Grief reminds us how profoundly capable we are to love. And just like love, grief is also non-negotiable and can show up quite unannounced. There is a quote I found online dedicated to a Jamie Anderson, and it goes, "Grief, I've learned, is really just love. It's all the love you want to give but cannot. All of that unspent love gathers in the corners of your eyes, the lump in your throat, the hollows of your chest. Grief is just love with no place to go." Thank you for being here and for your courage in taking the risk of feeling exposed and vulnerable by clicking on this episode. As per usual, there is both an English and a Turkish version of this episode. The Turkish version is dedicated to those who lost their lives during a recent hotel fire in Kartalkaya and also the victims of an earthquake that happened exactly 2 years ago. This version in English is dedicated to Max Lenail, an extraordinary young man who lost his life in a hiking accident in San Diego on January 29th, 2021 at the age of 21. I've had the privilege of getting to know him through his family and friends, and I'm honored to witness how hard his family has been working to transform their grief into connection and legacy, being an admirable example for how even the deepest and the most unimaginable sorrow can become a source of love and meaning. Max was studying neurobiology at Brown University at the time of his passing. I hope to do his memory justice with an attempt at an episode that he might have listened to as a fellow scientist. Let's dive in. I resorted to Shakespeare to start us off. Probably not what you expected from this episode. But in his Macbeth, Act 4, Scene 3, Malcolm says, "Give sorrow words, the grief that does not speak knits up the o'erwrought heart and bids it break." This is a profoundly accurate quote for real life. It means that unspoken grief or sorrow can weigh so heavily on the heart that it feels unbearable, leading to emotional or psychological collapse. By giving sorrow words, Malcolm is advising that expressing one's grief through speaking or mourning or sharing feelings can help ease the burden and prevent the pain from becoming overwhelming. As modern psychology would agree, it suggests that verbalizing grief is a necessary step for healing, and suppressing it can lead to greater suffering. Grief is not just a feeling, it's an unraveling. When we lose someone we love, the pain can feel like it's breaking us apart from the inside out. It's heavy, raw, and all-encompassing. And if you're in the middle of it, you might wonder, why does it hurt this much? Why does it feel like my body and mind are betraying me, weighing so heavily on me? Why does the grief feel like it physically hurts? The answer lies in neuroscience and biology, because just like all emotions, grief is also just a biological process. It impacts the very wiring of our brains, the rhythms of our hearts, and even the way we perceive the world. In today's episode, we'll explore the science behind this pain, looking at the acute and chronic phases of grief, why it's so physically and emotionally overwhelming, and how recovery might be possible. The best way to understand a human process is usually by looking into nature to find clues in other living beings. Grief, as we know it, is a deeply human thing, an ache in the chest, a quiet longing, the echo left behind when something we cherished is lost. No one escapes it. It's as universal as breathing. And yet, despite its deeply personal nature, grief is not unique to humans. It stretches beyond us into the animal kingdom and even into the forests or the microbial world. A tree in a dense forest does not have a beating heart, but it's not alone. Its roots tangle with those of its neighbors, speaking in the silent language of fungi and nutrients. When an old tree falls, the ones nearby don't simply carry on unchanged. They slow their growth, shifting their resources, as if they're mourning for a companion they can no longer reach. Microbes also deserve an honorary mention here. A single bacterium is small and unfeeling, but together they form communities, biofilms, colonies, networks. They share resources, they protect one another, and when catastrophe strikes, they change. When antibiotics wipe out half a colony, the survivors adapt and they send desperate chemical signals as if they're searching for those who are gone. Even plants hanging out still and silent seem to remember things. A mimosa pudica or pudica, not really sure how to say it, better known as the sensitive plant, will recoil at the touch of a hand. But over time, it learns which touches are harmless, and it stores the memory of past harms, responding differently in the future. If a plant can remember distress, if a tree can react to the loss of its kin, and if bacteria can change when their world is shattered, perhaps grief is not so much an emotion, but a fundamental rhythm of life itself. Maybe grief in its most primal form is simply what happens when a living thing loses part of what made it whole. And maybe, in ways we are only beginning to understand, the rest of nature has known this all along. This universality of grief is what allows us to turn to animals to understand our own pain. Through them, we learn that grief is not just an abstract sadness, but a deeply biological process, one that has shaped life itself. And by understanding it, we may find better ways to heal. And when we say grief research, the animals that come to mind are the prairie voles, a hamster-sized rodent native to Central and North America. What makes them so special is that they're monogamous. They form lifelong pair bonds with their partners, sharing nests and raising pups together, and even comforting each other during stress. This makes them an ideal model for studying social bonding, and by extension, the pain of losing that bond. A key study is from Dr. Larry Young and his team at Emory University, where they looked into the nucleus accumbens of prairie voles. Before explaining their findings, we need a quick intro to the nucleus accumbens. Nucleus accumbens is located deep within the brain in the ventral striatum, and it's a part of the mesolimbic dopamine system. When we engage in rewarding activities, anywhere from eating, socializing, to falling in love, dopamine floods this area, which reinforces that behavior. And the end result is not just about pleasure, but it's actually more about wanting and craving and seeking. When dopamine binds its receptors in the nucleus accumbens, it's more of a motivational hormone rather than a pleasure or happiness hormone. It creates the, oh, that felt good, and I want to feel that again sensation. Another very important molecule that binds this region is oxytocin, which we know is very important in pair bonding and also parent-child bonding after birth. It also allows for milk letdown from breasts, but that's beyond our topic. What we care about with oxytocin is that social or physical contact triggers its release into the nucleus accumbens to strengthen the reward association with the partner, and also into the hippocampus to enhance the memory of positive experiences with that partner. It works together with dopamine to make the interaction feel rewarding, and this dynamic reinforces monogamous bonds because the repeated bonding strengthens the neural circuits that make a loved one's presence rewarding. It also serves an evolutionary advantage because pair bonding increases the likelihood of caregiving and protection across generations. These interactions make the nucleus accumbens an obvious target to study bonding and love, and as their natural extension, grief. Researchers paired male and female prairie voles and allowed them to bond. Then they examined their brains and compared them to voles that hadn't bonded. In the nucleus accumbens of the bonded voles, there was an increased expression of oxytocin receptors. Receptors and hormones are like a door and a key. More receptors means more entry of oxytocin. This means pair bonding had caused epigenetic changes in the nucleus accumbens, which are chemical modifications to DNA that can turn genes on or off. In response to the high oxytocin release, the DNA had allowed for more doors for entry. Follow-up studies also looked at what happens when voles are separated after bonding, and they compared monogamous voles with non-monogamous voles. After separation, the monogamous voles showed distress. They called out for their partners, tried many ways to get to them, and even had higher levels of the stress hormone cortisol. And their brains also showed less oxytocin binding in the nucleus accumbens. The loss of their partner had disrupted their brain's oxytocin circuitry, making their separation harder to cope with. Essentially, their brains were wired to seek out their partner, and when that partner was gone, it left a kind of neurological void. And the non-monogamous voles, who formed weaker bonds and showed less distress when separated, showed that they had fewer oxytocin receptors to begin with. The number of receptors that their DNA determined correlated with the strength of their bonds and their grieving. And before you get ready to blame everything on your oxytocin receptors in case you're not very monogamous, a study that genetically increased oxytocin receptors in the nucleus accumbens did not alter the meadow vole's social preference behavior. So there are more factors than oxytocin at play. Maybe that's for another episode. Studies like these naturally led to human studies looking at the nucleus accumbens. Functional MRI advancements are very key here. It's like getting an MRI, except the algorithm measures real-time activation of brain regions by following where hydrogen flows. They studied people who are grieving for a loved one, and when they were asked to think about the deceased person, the nucleus accumbens lit up with activity. And the more intense their grief was, the more active this region became. This is a good place to distinguish between complicated and non-complicated grief. Most people, as painful or slow their process might be, eventually learn to live without the person they lost. The nucleus accumbens adjusts and our brain's attachment system rewires itself. But for 10 to 20% of people, grief doesn't ease up. It stays raw, all-consuming, even years later. And this is called complicated grief. The nucleus accumbens stays stuck in a loop almost. In non-complicated grief, the brain reduces its dopamine responses to the lost one. But in complicated grief, the nucleus accumbens keeps firing, reinforcing the craving. The brain is still expecting reunion, as if the person is about to walk through the door, even though the conscious mind knows that it's impossible. Oxytocin also doesn't adjust properly, keeping the brain stuck in attachment mode. And stress hormones also stay high. There's actually a valid predictor for whether you are more inclined for complicated grief or non-complicated grief. And that's your catecholamine levels: epinephrine, norepinephrine, and dopamine. You'll recognize epinephrine and norepinephrine, also called adrenaline and noradrenaline, as the regulators of your fight or flight response to stress. Your body has crucial mechanisms in place to balance your sympathetic and parasympathetic nervous systems. A stressor stimulates the sympathetic system, and epinephrine, norepinephrine, and dopamine, they all work together to increase blood flow to your muscles and brain, to increase alertness, increase heart rate, and blood pressure. Your pupils get bigger so that you can see more light. Your skin becomes pale because blood is diverted from the skin to the muscles, so that you can use the muscles to run away. Your heart beats faster, you breathe faster, and you use more glucose for instantaneous energy. All of this is balanced by the parasympathetic system, which acts as the brake and turns on the rest and digest mode. It slows your heart rate and blood pressure. It tells your brain that you're no longer fighting or running away from threat, that it's safe to rest. Studies show that your levels of these catecholamines can predict how you'll react to grief once you lose someone or something, and they can also predict your treatment response while grieving. Higher levels of autonomic arousal, as determined by our catecholamines, allow for a higher baseline of anxiety and alertness. And this correlates with higher odds of prolonged, complicated grief, whether you're seeking professional help or not. So, the state of our catecholamine balance can determine how you show up to grieving and how you go through it. Higher sympathetic nervous system activation also increases systemic inflammation. Your immune cells react to norepinephrine by producing pro-inflammatory cytokines like IL-6 or TNF-alpha. Your hypothalamic pituitary adrenal axis, the HPA axis, also gets activated, and this increases cortisol. Initially, cortisol helps regulate inflammation, and it's an essential hormone for your everyday functioning. But when it's chronically high, the immune cells become less responsive to its anti-inflammatory effects, and this causes persistent low-grade inflammation, which can be seen in prolonged grief, depression, or PTSD. There are many studies that show changes in inflammatory markers and stress markers in blood of those living with these conditions. Speaking of depression, even though there certainly are common symptoms between depression and grief, these are biologically different processes. Depression is a disorder of mood, and grief is a response to loss, and it's a motivational state with a desire for the thing you lost. This is why antidepressants don't really work for grief. They increase serotonin, which can certainly help with depression, but they don't fix the hyperactivity of the nucleus accumbens and the yearning seen in grief. The predictive link I mentioned earlier with catecholamine levels is also not seen in depression, only in grief. So while grief and depression often coexist, they are biologically different. Next up, I'll talk about neural circuits responsible for identifying whether something is present or absent. Imagine you're driving home on the familiar road. You don't even have to think about where to turn. Your brain just knows where to go. But then, one day you hit a dead end. The road is gone. And that's what happens in grief. Your brain has spent years mapping out a world where your loved one exists, where they sit at the dinner table, where their voice sounds in a crowded room. But now they're gone, and your brain doesn't know what to do with the empty space. You're confused and disoriented because the nucleus accumbens loses its balance. And there are neural circuits in the hippocampus that influence what's happening in the nucleus accumbens. If you watched episode 3, you know all about the role of the hippocampus in memory, in spatial awareness, and also contextual memories. Your brain has thousands of different types of neurons that are each subspecialized. It's not much different at all from how an orchestra operates. If you have an orchestra devoted to the hippocampus, when performing something about grief, for example, your main instruments are place cells, proximity cells, and trace cells. The names are pretty self-explanatory. Place cells fire when you're in a specific location. They create a mental map of your environment. If you're in your kitchen, place cells that are encoded for the kitchen fire. Proximity cells fire based on how close you are to something or someone. They map both physical distance and emotional distance, like how far you are from your front door or how close you are to a loved one. It's an internal GPS. Say you're sitting next to your loved one and your proximity cells are firing to create a sense of closeness and connection. And this particular activity has a great role in stimulating oxytocin release. And being close to someone you love becomes rewarding and intoxicating. Even when you become physically apart, these cells can still fire when you think about that person, and this way creating a sense of connection despite the physical distance. Next are the trace cells, which are for remembering sequences of events. They help you link memories across time. And they've been found to encode the absence of expected stimuli or events, especially when there is a temporal gap between stimuli. They're thought to be maintaining a representation of a stimulus during a delayed period after it has disappeared. When you walk into a room where you expect to see your loved one, your trace cells fire in response to their absence, creating a longing or yearning. The brain is fascinating in its ability to anticipate and respond to expected stimuli. And this ability makes it very sensitive to when a learned stimulus has to change. fMRI studies show the hippocampus to be highly active during grief, especially when participants are asked to recall memories. Again, in the third episode, I explained that the hippocampus only stores short-term memories. They then get stored in the cortex to become long-term memories. So the increased activation here in grief seems to be not so much around remembering long-term memories or making new memories, but more so about recalling the context of these memories, where you were, what you were doing, and how it felt to be with them. During grief, the hippocampus is retrieving the context of memories while processing the absence of expected stimuli and linking the emotional and contextual aspects. Place cells remind you of the memories tied to each place with that person. Proximity cells remind you of their absence, and trace cells remind you where your loved one should be and the sequence of events that should be unfolding but are not. It's a neural dissonance. Your brain is constantly trying to reconcile the presence of memories with the absence of the person, trying to recreate the context while constantly reminded of the painful reality of their absence. It's because of this neural circuit that grief is so disorienting. You turn your head expecting to see them in a familiar place, or you feel their presence in a room, even though logically you know that they are gone. Or certain locations become overwhelmingly painful because your brain is wired to expect them in those places. And it simply needs time to rewire. It's this hippocampal circuitry that tells the nucleus accumbens that the person should still be there. And remember, the nucleus accumbens is like your brain's GPS for reward. It learns what's important, locks it into memory, and anticipates future experiences with it. When it doesn't get what it expects, like in grief, it freaks out. It keeps recalculating. It keeps searching and craving. It's like when your GPS refuses to accept that a road is closed. It keeps redirecting you back, trying to make the old route work. This is what makes grief feel so raw. The nucleus accumbens keeps sending dopamine signals, driving the feeling of intense yearning, but there's no reward. The hippocampus keeps lighting up the maps that once made sense, but the landmarks are gone. The entire system gets stuck in a loop of expectation, absence, and pain. All of this might make grief sound like it's impossible to recover from, which isn't true. Maybe one of the first realizations has to be that healing from grief doesn't mean forgetting. It means allowing the brain to update its maps. It means helping the hippocampus adjust its place, trace, and proximity signals so that familiar places no longer feel like empty, haunted places. It also means helping the nucleus accumbens relearn where the joy can come from. This is why social support and new experiences and even movement-based activities help people recover. They give the nucleus accumbens new things to look forward to and help the hippocampus rewrite the maps. Because in grief, the road doesn't disappear, it just needs to be redrawn. And it's completely natural from a neurobiological perspective that everyone grieves differently, and there is rarely a need to pathologize it. You're not broken for grieving the way that you do. It's how you are wired. And we have evidence-based tools that can help you navigate this process of healing. Some already well-known methods are engaging in new activities, meditation, exercise, all of which contribute to neuroplasticity. Allowing yourself to feel and process your emotions by journaling or therapy, or putting your emotions into art and creativity, and leaning on your social support system, creating memorials. These have all been shown to be incredibly helpful. A method you may or may not have heard of is increasing your vagal tone, which is actually part of any meditation or other calming activities that you pursue. Your vagus nerve is the longest cranial nerve in your body, and it's a key player in your parasympathetic nervous system. It connects to many parts in the body to regulate your heart rate, breathing, and even your emotional responses. Vagal tone is the activity level of this nerve. Higher vagal tone is associated with better emotional regulation, lower stress levels, and faster recovery from emotional setbacks. Think of it as your body's natural resilience system. When you're grieving, your body is under a lot of stress. Your sympathetic nervous system is often overactive, leading to symptoms like a racing heart, shallow breathing, and difficulty with sleeping. This is where the vagus nerve comes in. A strong vagal tone helps activate the parasympathetic nervous system, calming your body and mind. Research has shown that people with higher vagal tone tend to recover more quickly from grief and trauma. They're better able to regulate their emotions and process their feelings. And increasing your vagal tone is easier than you might think. Here I will highlight the role of the inhale-exhale balance. A marker of your vagal tone is respiratory sinus arrhythmia, or RSA. This is the natural heart rate fluctuation in sync with your breathing. When you inhale, your lungs expand, your diaphragm contracts, and the pressure in your chest cavity decreases. This stimulates the sympathetic nervous system, and your heart rate slightly increases. When you exhale, your diaphragm relaxes, the chest cavity shrinks, and the pressure in the chest increases. This causes the parasympathetic nervous system to decrease your heart rate. Think of the times you inhale more strongly. When you're afraid, the first thing you do is inhale and you keep taking inhalations. The inhalation and the following pressure changes allow the heart to pump more blood to meet the increased oxygen demand. When you finally exhale, you find yourself relaxed and it calms you down. So next time you're anxious, try focusing on exhaling properly rather than taking a lot of consecutive breaths in, which, now that you think of it, probably only makes your anxiety worse and makes you dizzy. RSA is this rhythmic variation in heart rate because of the back and forth between inhales and exhales. Higher RSA means greater variability and better vagal tone, and it's associated, like I said, with better emotional regulation and improved physical health. Lower RSA is often seen with chronic stress, anxiety, or depression. Improving your RSA can be as simple as doing slow, deep breathing with longer exhales than inhales. Try inhaling for 4 seconds, holding for 4 seconds, and exhaling for 6 seconds, for example. Meditation and mindfulness practices that focus on breath awareness can also increase RSA over time. And so can singing and chanting, moderate aerobic exercise, and even cold exposure, like when you splash your face with cold water. Another crucial factor in balancing the autonomic nervous system is light exposure. Natural daylight, especially in the morning, helps regulate cortisol rhythms. Excessive screen time at night, on the other hand, suppresses melatonin production and disrupts the natural rise and fall of cortisol, potentially exacerbating stress and emotional dysregulation. Strategic light exposure, getting sunlight early in the day and minimizing blue light from screens in the evening, can help stabilize the nervous system, promoting a sense of calm and aiding in grief recovery. In the context of grief, it's been shown that those with increased vagal tone and thus higher RSA may get more benefit from healing exercises, such as written disclosure or grief journaling. Whether you're grieving or not, increasing your vagal tone is a great exercise for overall health and to help regulate your stress hormone levels. There is one more concept I want to touch on, and that's collective grieving, the shared experience of grief that occurs when a community, society, or even an entire nation mourns together. I mentioned that the Turkish version of this episode would be dedicated to the earthquake and fire victims. My family and close friends thankfully didn't lose a loved one in these disasters, but we grieved. We were absolutely heartbroken. When the earthquake happened, where more than 50,000 people died, I wasn't even in Turkey, and I couldn't sleep for days. I was constantly watching the news for updates. I was crying nonstop at every social media post, feeling as if the buildings around me in Toronto could crumble down any second. And I kept wondering, If I were in that state, how were those directly affected by it doing? And for the purposes of this episode, why do we grieve for people we've never met? At the heart of this is empathy, rooted in a network of brain regions, including the anterior cingulate cortex and the insula, which helps us process emotional pain, both our own and others. A key player in empathy is a group of neurons called mirror neurons. They fire both when we perform an action and when we see someone else perform the same action. You see someone smile, your mirror neurons fire as if you were smiling too. So when we see images or hear stories of people suffering after an event, our mirror neurons fire as if we were experiencing that suffering ourselves, and we feel a deep emotional response. It's not just sympathy, it's a visceral neurological reaction. Another factor for collective grieving is also our innate need for social bonding and our sense of belonging to a tribe or a community. Humans are inherently social creatures, and our survival has always depended on our ability to form strong group bonds. And this makes us feel connected to others, even if we've never met them. When a tragedy strikes, it often feels like an attack on our tribe, whether that's our local community, our nation, or even humanity as a whole. This triggers a deep psychological response rooted in our evolutionary history. We mourn not just for the individuals who were lost, but for the sense of safety and unity that's been disrupted. There is also the additional factor of shared identities. When we identify with a group, whether it's based on nationality, religion, or shared values, our brains treat that group as an extension of ourselves. This is why we feel a sense of loss when members of our group suffer or die. Studies have shown that when we think about our group identity, brain regions like the medial prefrontal cortex become active. This area is involved in self-referential thinking, basically how we see ourselves in relation to others. When we mourn for strangers, it's because our brains have integrated them into our sense of self. For example, after a natural disaster, we might see images of people who look like us, live like us, or share our values. And this activates our medial prefrontal cortex, making us feel like their loss is our loss. Ernest Becker, in his Pulitzer Prize-winning book, The Denial of Death, argues that much of human life is spent suppressing thoughts of mortality and loss. Death is too vast, too incomprehensible for us to confront directly, so we push it aside until grief forces it to the surface. He writes, "Modern man is drinking and drugging himself out of awareness, or he spends his time shopping, which is the same thing. As awareness calls for types of heroic dedication that his culture no longer provides, he is brought to his knees by the reality of death. Grief, then, becomes not only a reaction to the loss of someone, but also a confrontation with our own finiteness. It's an experience that makes us ask big questions about existence, identity, and purpose. Grief is not just a pain to endure, but a window into the depths of who we are. It strips away pretense, leaving us raw and exposed to the reality of love, loss, and the impermanence of life. As I come closer to the end, let us sit with the truth that grief is a testament to love. The pain we feel is the echo of the love we gave and received. It's a proof that a connection so deep cannot simply just disappear. Grief is not just an ache, it's a fierce reminder of what once lit up our lives. And it's up to us to determine how the connections we shared continue to shape us in ways that endure beyond their physical presence. I will once again resort to Shakespeare, to lines not originally written in the context of grief, but I believe to be applicable and have helped me reflect on grief. In Romeo and Juliet, Act 3, Scene 2, Juliet is eagerly awaiting nightfall so she can reunite with Romeo, unaware of tragedies yet to unfold, and says, "When he shall die, Take him and cut him out in little stars, and he will make the face of heaven so fine that all the world will be in love with night, and pay no worship to the garish sun. To grieve is to love, and to love is to keep the light of those we've lost alive, even in the darkest nights. Grief transforms the way we see the world. It turns ordinary moments into sacred ones, night times into times of reflection, and loss into an opportunity to create something lasting. Max's family not only held on to and strengthened their social bonds and created a community dedicated to honoring the light Max brought into this world, they have also put together an immense project to build a bridge where he was lost during his attempt to cross the water. A bridge to ensure no one has to ever risk their lives to complete that hiking trail. Just as Max's family turned their loss into connection and remembrance, we too can honor the ones we miss by building bridges between memory and meaning, between sorrow and hope. So tonight, when you look up at the stars, perhaps you'll see not just light, but a tapestry of the connections we have lost, yet still hold dear. And in doing so, may we all find a little peace, a little hope, and the courage to keep building bridges, guided by the enduring light of those who no longer walk beside us, but continue to be a reminder that love outlasts even death. Thank you for joining me today with both your scientific curiosity and emotional vulnerability. Remember to honor your grief, speak its fruits, and let the love it carries shape the stories that you continue to tell. Let that love forward into the world. Santé.